potential that is maintained by the cell, we actually use out a lot of energies. They use a lot of ATPs. So the food that we take in mostly is to generate energies. There's the ATP in the cell by the mitochondria. And from there, it's able to basically maintain the membrane potential so that it can communicate between cells. Either they are excitable or non-excitable cell. But the difference between excitable cell, because it has a voltage-gated ion channels that is present on the cell membrane, and that is very important for excitable cells. And we know that the excitable cell are the neurons, which we have looked at in the synapse yesterday. Oh no, a day on Tuesday, yeah. And then today we are going to look at the muscles, okay. And in this course, we are going to look only two, basically the skeletal muscle, and on Monday we are going to look at the smooth muscle. Okay, so skeletal muscle, smooth muscle, and the cardiac muscle, they are basically an effectors. Okay, so they are the effectors. Yeah, so they are the one that basically helps us to be able to basically respond after the stimulus is applied. So the primary function of the muscle is to basically generate forces or movement in response to physiological stimulus, which means that there is any stimulus that is physiologically and the cell is able to interpret in the control center, it will be sent to the effectors. So the neuron are basically the transmitters line, okay? The transmitters line, they're basically able to transmit the action potential, which is basically the electrical signals to the effectors so that the effector can respond. And we know that the primary function of muscles is basically to generate force. Okay, so now we are going to look today how the skeletal muscle is able to generate the force or the movement that is required in response to the physiological stimulus. Yeah, and we know in our human body we basically has three different types of muscles. They are adapted to specialized function. Yeah. So this is basically the brief overview on that, and then we are going to look at the molecular mechanism of the contraction and how contraction relaxation happens in the skeletal muscle. So we look at the learning outcome for today's class. There is basically one, to compare and contrast the three different type of muscles that is present in our body. Okay, is there any other than the three types of muscles that is present in our body? So basically the answer is no, it's only the three types. So with the help of a drawing, you will describe the gross structure, look at microscopic structure and how the molecular structure of the skeletal muscles. Yeah. Then we look at the mechanism of contraction and also then the relaxation. Okay. And what happens when the muscles is at rest, and how much energy that is basically required during exercise, and compared to the three type of muscle fiber that is present in the muscle bundles. Okay, and discuss the significance of slow motor unit. Then we look at how the motor unit actually can be subdivided into three different types. There's the slow type, the fatigable type, and also the fast fatigue resistant motor unit. And then we look at the disorder in the muscles. So these are the 7 learning outcomes we hope we are able to discover or uncover, that you are able to uncover and find the knowledge to add on to your knowledge that you already probably have. But it will be more details looking at the molecular structure and how it actually helps the muscle to contract. Okay, so the first learning activity we're going to look into is basically 11.11. Then you can turn to page 7, looking at how the muscle help to maintain and changing posture, how it help the limbs to move. And basically when we speak to our friends, we show some sort of facial expression and also speech, and the movement of the internal environment, internal organs, such as basically the contraction of the heart, the lung, how does it expand, then urination. But these are more specific systems that we are going to do it when you do a systemic block later on in your, starting from your second semesters, looking at cardiovascular system and also on the respiratory system. Yeah. Then movement of food, that's basically the GIT, our gastrointestinal tract, looking at movement of food through our GIT. Yeah. Okay, so these are basically the main important function of the muscle. As I said just now, that the primary function of muscle is to generate force or movement in response to physiological stimulus. Yeah. So you look at activity 11.1, looking what is the specialized function of the muscles. Okay, so when we talk about muscles, then you basically have to see what type of muscles they were talking about, and the reaction enable the muscle to contract. Okay, so actually these hints that provide you is basically to guide you to uncover the questions that is being asked. Okay, so hint says that muscle cells contain contractile protein that are activated during contraction. So you can basically answer that. I just want to look at the, okay, yeah. We basically know in this, looking at the skeletal muscle, okay, it's basically composed of almost 40 to 50% of our body mass. As I mentioned, that if the whole body that we have, most of it is basically made up of skeletal muscles. There's about 50% or more actually. For those who are thin, probably they have more muscles compared to those who are putting on weight, then you have slightly less muscle mass. Yeah. So compared to the men and also to the ladies, the men probably have more muscle mass. Okay, that is also due to their hormones because of testosterone. So you need to, if you look at the evolutionary aspect, basically men need to fight for food and need to go out and work for food. So your muscle mass tend to be bigger, and compared to women, they will be slightly less. Yeah. So between 40 to 50%. So we can safely say is that 40% probably refer to the ladies and 50% will refer to the men. Yeah. So the contraction and activation of the muscle fiber within a tendency of muscle to shorten. So we know that basically if there is a contraction, so how does that actually helps? So in all the muscle contraction, it must be due to the increase in cytosolic calcium concentration. Okay, so there must be a channels that basically opens that allow calcium to either get in from the internal environment or get in from the intracellular aspect, meaning released from the organelles. One of them is the sarcoplasmic reticulum. Okay. And we know that basically skeletal muscle is important for them to basically help to maintain stability and also power for all our body movement. And any problems with that, especially when you see a person who has stroke, then you see there will be an instability or immobility. Yeah, if there's an impairment. So you can work on activity 11.1. So we have basically got you through looking at answer the first one. And then you can list down as many examples as possible for that illustrate the significance of muscle contractions, comparing the smooth muscle and the cardiac muscle. Okay, so can you give me a list of activities by the contraction of the muscles? Examples. The significance of skeletal muscle contraction, or smooth muscle contraction, or cardiac muscle contraction. Which one do you want to say or want to give an example? Can someone respond?
Yeah, doctor.
Yes, please.
For skeletal muscles,
Yes.
like your respiratory muscles, so the ventilation.
Okay, that's good. So for skeletal muscle, basically our diaphragm, when it contracts, it helps volume, which basically reduce the pressure and allow us to basically undergo having this ventilation. That's very good. Anyone want to give any other examples?
When it comes to the cardiac muscles,
Yes.
the contraction changes the size of the heart chambers.
Okay. Sorry. Okay. So after breaking halfway through, I don't know, probably I didn't listen. I don't know about the rest. So because basically when this heart is contracting, it changes the size of the chamber. That's what it means. The most important thing is he is able to basically push or create pressure. He is able to squeeze the blood out from the heart. So this is what we call the cardiac output. Yeah. So the stroke volume that the heart able to generate is basically pushed into the aorta. Okay, that increase in volume basically increase the pressure that allow the blood to move from high pressure to low pressure, which means from the aorta going down to the artery, arterial, capillaries, and finally get into the venule, okay, and then the veins, and then finally get back to the heart again, the right chamber. Yeah. So that is the activity that it can help basically to move. So they are very significant without the heart contracting, without stopping since the day we are formed until the last of our minute or seconds that we of our life, it will be contracting. Okay. Any other examples that you want to give? How about smooth muscle?
For the
Yes.
smooth muscles to the peristalsis of the GIT, doctor.
Yes, peristalsis of the GIT. So the significance is once the GIT able to peristalsis, basically able to move, it help to propel the food forward, starting from our esophagus going down. The time we swallow, after that is basically up to the GIT to move the food that we ingest. Okay, and the food bolus basically travels throughout the length of the GIT. Without that movement, what will be the consequences? If let's say the GIT decided, the sumatra decided not to contract. What will be the, yes?
The person will be choked.
Yeah, probably he will be choked, but let's say the food able to get down. Because the first 1/3 of our esophagus, we are able to basically still control it. But once the food get to the second third of the esophagus, basically it's up to the GIT. So probably the food was stuck in the GIT, I mean in the esophagus. But as you go further down, if let's say the large intestine decided not to move, that is where you are going to get constipation. Yeah. And it's not going to help in digestion. So there will be a lot of things that actually happens if the muscle decided to have stop working or there are problems there. So if any impairment, that is the main problem. I mean, the same thing for the heart. If the heart decided not to work, that would be basically cardiac arrest, and it's going to cause death. Yeah. Okay, same thing. If the skeletal muscle doesn't work, then we are going to be immobilized or paralyzed. Okay, so you can list down as many as you go on, and then from there you can see basically they are very significant. So I repeat again that the main function of our skeletal our muscles is mainly, primary functions is mainly to generate force or movement in response to physiological stimulus. Okay, so we can go on that and then explains how muscle contract can bring about the activity that we have listed. So the skeletal muscle, the most important thing, it must have a point of insertion on our bone. And usually our skeletal muscle works in pair, okay, and most of the time they have to be in pair, okay, except the diaphragm, which is still a single one. The rest of it mostly for movement, usually they work in pairs. Yeah. So we basically have done activity 11.1. If you have any questions, if not, I will just move on to the next one, looking at the gross and the histological aspect of the skeletal muscle. Okay. Okay, for those who have done, if not, we just move on. Then 11.2, gross activity, we're looking at the bicep, tricep, and then the tendon is the insertion. So basically, you look at the skeletal muscle. I think the rest of that, basically, you are able to explain. Yeah, looking at the skeletal muscle. And then we look at activity. Why are the skeletal muscles striated?
The presence of actin and myosin.
Okay, because of the contractile protein that is arranged in a sequence or systematically arranged, so you can basically view the skeletal muscle grossly, basically histologically, you can basically see them that they are well arranged in systematical order. So they are basically seeing stripes, just like the zebra, but the zebra stripe is not that systematically arranged, but you still can see black and white. Yeah. So the same thing as the skeletal muscle, they are more like a stripe. So you can see the black and the white, the stripes. Yeah. So what is the most abundant constituents of the skeletal muscle fiber? Okay, so most of it are basically the contractile protein. And we know now basically the contractile protein consists of the thick and the thin filament. Yeah. And in the thin filament, you have all this, the actin, the myosin, the actin, the troponin, and tropomyosin. Okay, so how do skeletal muscle enlarge with appropriate exercise? Does your skeletal muscle actually increase in number of cells?
Yes.
Okay, yes, okay. If it do, then you are going to get more and more muscle cell, especially those people who do bodybuilding or you go for running. So your gastrocnemius muscle basically enlarge. In actual fact, it hypertrophy. Okay, that enlargement is not actually due to the number of skeletal muscle cell or that is becoming more, because when we are born, the amount of skeletal muscles or cell that is available is fixed. Okay, so the only thing that make it difference is become bigger. So it hypertrophy, yeah, increase in size. It's not increase in number, yeah. Okay, so basically enlarge by increasing in size. So the muscles mass become bigger. That's where all the contractile protein become more and more as the person engage in an appropriate exercise like bodybuilding and some sprinters or swimmers. So this is the areas where you can build up your muscles by increasing in size. Okay, so now how is skeletal muscle contraction regulated? Which means the question is, can the muscle contract without innervation, without nerve supply?
No, doctor.
Okay, why skeletal muscle cannot contract without nerve supply? Whereas compared to smooth muscle, it can contract, or even the cardiac muscle can contract without nerve supply. Okay, this will be a good thing for us to explore then. So which means that if we put the cardiac muscle in a solution free of calcium with the intact nerve, will the muscle contract compared to if you put a smooth muscle in a solution free of calcium? Will the smooth muscle contract? Okay, so you are going to uncover this. So in the meantime, think about it. Yeah, and we are going to explore the next few days. That means today and also on Monday about smooth muscle. Yeah. So how is skeletal muscle contract? Basically, it's innervated by the somatic nerve, and we know the somatic nerve are basically the motor nerve. So you have basically the nerve that is the efferent nerve that is coming down from our central nervous system. Okay, the spinothalamic tract basically is the upper motor neuron coming down to your lower motor neuron. From there, it innervates directly with the muscles. At the point of contact with the muscles, the nerve with the muscles, there is a junction which is known as the neuromuscular junction, and we know that there is the synapse, yeah, where the muscles is contracting, and we know there's a synapse. We have been exploring on Tuesday a lot about the synapse and how it actually works in the synapse, yeah. Okay, but for skeletal muscle, the cell membranes have the voltage gated channels. Okay, since we say that if there's a voltage gated channels, it must be able to transmit information. Compared to the nerve or the neuron, the voltage gated channels that is present almost exclusively to the axon, the whole length of the axon, yeah, the whole length of it. The main purpose is to transmit calcium potential, okay, through another nerve or through another cell, so it can actually transmit the information. But for the skeletal muscle, the presence of voltage gate on the cell membrane is mainly to allow the cell to only basically transmit information to itself, not to another person or to another cell, or spread or send the information or the message. So the information that it has is basically only for itself. So it's going to be transmitted down through the T tubule to basically activate the calcium L-type calcium gate to be open. Okay, then basically release the calcium into the internal environment of the master cell. Yeah. So we are done with that, unless you have questions on activity 11.2. No. Okay. So you can also look at the gross structure of the smooth muscle. Okay. So I think you can actually label that looking at the tunica, the blood vessels. So you can draw a blood vessels, which is in the diagram figure 11.2a. Okay, then you can label them accordingly. So what is the implication of smooth muscle contractions in the arterioles? So we know that the whole length of the smooth muscle of the blood vessel from the aorta down the arteries, okay, finally going to the arterioles before it actually enter into the capillaries. So what is the implication when the smooth muscle at the arterioles start to contract? Any idea?
They help regulate blood pressure.
Yes, basically they regulate the blood flow to the capillaries. By constricting the blood flow to the capillaries, now the blood pressure in the artery basically increase. So if we have vasoconstriction, basically we are referring to the constriction of the arterioles. That basically helps to increase the total peripheral resistance. That indirectly basically cause the blood pressure to increase because the diameter of the blood vessels reduce. Okay, that increase the blood pressure. Yeah. So if there is a problem with the arterioles or they are basically overstimulated, that's why people have hypertension. Yeah. Or it can also be due to volume of blood or ACF that we have discussed earlier. But meaning if the arterioles is constricted, the blood pressure will increase because it increases the total peripheral resistance. Okay. Now, what is the consequence of smooth muscle contraction and relaxation? Yeah. So we have discussed just now about peristalsis. We talked with Ali was talking about the GIT, so that's important. Yeah. And then you can actually go to all the examples that is given there, basically urination, starting from defecation, childbirth, and also the vasoconstriction, which we have discussed just now on the arterioles. Yeah. So for 11.2b, figure shows the longitudinal section of the smooth muscle, and we know that basically there is no strike in the smooth muscle. So if you look at the smooth muscle, basically there is no strike. Okay, how is it able to organize itself? Yeah, and how is able to basically contract without the stripes? Okay, so they are smooth. Yeah, so you can see in figure 2b, they are smooth and they are spindle shaped. Okay, but they are basically connect to each other with the gap junction. Okay, so we are going to explore that again more, and then how is smooth muscle regulated? So basically, smooth muscle is regulated by the autonomic nervous system, not the somatic nervous system. Yeah. Is that okay? And also the hormonal system, basically the chemicals. So will the smooth muscle contract in an environment free of calcium? Then you look at how the hormones actually help the smooth muscle to contract without a nerve innervation. Yeah. Okay, so we have done activity 11.3. Today there's not much drawing. So we're looking at more on how to basically uncover the activities that is here. That I hope that if you have done, then we are going to do that. If not, we can go in together. So looking at 11.4, the gross structure of the cardiac muscles. Okay. We look at figure 11.3a, look at cardiac muscle with Akka has just now mentioned about that. The contraction basically reduce the chamber. So we can basically view how the heart actually look like. Okay. Then what is the implication of the muscle cardiac muscle contraction we have discussed just now? That it basically reduce the the chamber size that help to propel or increase the force or pressure in the chamber that basically push the blood out of the heart. Okay, and this is what we call stroke volume, and basically times the heart rate will give you the cardiac output. You are going to explore that when we do cardiovascular system. Yeah, and they also basically get junction like the smooth muscle, but they also have the stripes like the skeletal muscle. Yeah. So what is the consequences if the cardiac muscle have disorder? So what will be the consequences if there is a cardiac muscle disorder? And can you? Yes. Anyone want to try to respond? No.
What was the question, doctor?
The question is in the module 11.4. What is the consequences of cardiac muscle disorder?
They can't move properly.
Okay,
They
it can't
have
contract. Yeah, that's good. Okay.
Tachycardia.
Tachycardia, that means it increased too fast. So it's contracting too fast, or difficulty in contraction. So
if cardiac arrhythmia, doctor?
Cardiac arrhythmia, yeah, that can happen. So it's an uneven cardiac contraction that without the proper contraction, it can basically not able to pump a proper amount of blood out. So you probably can cause reduce in blood pressure because there will be less blood that's being pumped out. If tachycardia, there's too fast the heartbeat, also reduce the blood output, the stroke volume, because it has no time to rest, to break for filling up the heart chamber. So that also will reduce the stroke volume. Yeah, that basically can also reduce the blood pressure. So we know that the consequences of hypotension compared to if the person have a high blood pressure. So either way is not good. Yeah. So how is the cardiac muscle contraction regulated? Any idea? Mainly if we cut off the blood, the nerve supply to the heart, will the heart contract?
No, it will, doctor. It's myogenic, right?
Yeah, so it will be able to contract even if you cut off all the autonomic nervous supply, basically the sympathetic and the parasympathetic nerve supply to the heart. Okay, because it has its own pacemaker cell. Okay, that's what we call about pacemaker potential. So it can generate its own impulse, electrical signals that allow the muscle to contract in rhythm. Okay, and because it also connected to each other by the gap junction, so basically the muscle is able to contract and basically connect to each other, and that allow it to contract in synchrony. So even without the nerve supply, in actual fact, if there is no nerve supply or parasympathetic nerve is being cut, sympathetic nerve is being cut off, the heart rate actually increase from 70 to 100. Okay, which means that the pacemaker is able to generate a speed of contraction of about hundreds per minute. Yeah. So how come we are able to have a heartbeat of only about 70 to 75 or 77? So there must be some sort of nerve regulation that actually reduce the heart rate. Okay. And we know that the heart is only innervated by the autonomic nervous system. That is the sympathetic and the parasympathetic. Okay, so it is the parasympathetic that is actually reduce the heart rate from hundreds to 70 or 77. Yeah. So the increased activity of the parasympathetic actually reduce the heart rate to 70 beats per minute. So if we inhibit the parasympathetic nerve, then the heart rate will increase to 100. Yeah. That even we don't even need to activate the sympathetic nerve. Okay. What happens if you activate the sympathetic nerve? Then the heart rate will increase to more than 100, 110, 120. But as it go more than 120, basically it start to go into tachycardia. Yeah, that is not a good sign. Okay, so that's all about the activity 11.4. If you have any questions, is that okay, Ani?
Yes, doctor.
Yeah, okay. So we move on to the next one, type of cardiac muscle, and you look at table 11.1. There is basically comparison between the skeletal muscle, smooth muscle, and the cardiac muscle. Yeah. So we already know now basically skeletal muscle are striated, cardiac muscle is in between stride. Okay, it also have stride but with gap junction, and smooth muscle is smooth. There's not stride, but it also connected by gap junction. Yeah. And only skeletal muscle is voluntarily, whereas smooth muscle and cardiac muscle, they are involuntarily. So why is we call smooth muscle a voluntary smooth muscle? Sorry, the skeletal muscle. Why we call them a voluntary skeletal muscle? Anyone? Yes,
Because
sorry, Yawaka.
sorry. Okay. Because we can move them under like conscious control.
Okay. Why is that significant, Waka?
So that we can move willingly and even like protect ourselves from external stimuli.
Yeah, that's good. But the most important thing, the nerve must be innervating the skeletal muscle, and we know that that is a somatic nerve, which is a motor nerve, and is in our control. So the whole body system that we have currently is only one thing that we can control. That is the skeletal muscle. Most of the things that happens to us, is not under our control. Yeah, just imagine that if God or Allah decided that you can control everything in your body, I think you don't even have time to think about what to do next. Yeah. So to control the skeletal muscle, that is very important, as Wakar said, basically for us to be able to create movement and also to give us a posture. It means we are able to basically stand out, sit out, or basically sit straight. If some of you are still lying down or enjoying yourself, basically that is, if you want to go to the toilet, you can actually do that and move your skeletal muscle, and you can actually move. Yeah. So we basically has a control because it's innervated by the somatic nerve, which is basically the motor nerve from our central nervous system. Okay. So they are able to control. What happens if we can control the skeletal, the cardiac muscle? Then we are going to have problems because we don't actually know how many beats to basically get the heart on the right contraction. Yeah. And the same thing as smooth muscle, if we're going to think about contracting or controlling the smooth muscle, then we basically have no time to think about doing other things. Yeah. So we leave the smooth muscle to be involuntary, and the same thing for the cardiac muscle to be involuntary, so that basically it can beat throughout the whole of our life until our last second. Yeah. So if there is any dysfunction of this, okay, which type of muscle is most dangerous to the human? If there's this dysfunction of which type of muscle, look at the last questions. Which is most dangerous to us and why?
The cardiac muscle?
Yes, the cardiac muscle. Why we say if there's a problem with the cardiac muscle is most dangerous to us, not the skeletal muscle or the smooth muscle?
Because it will affect directly the heart.
Yes, it basically direct affect the heart, and we know that controlling the blood pressure is the most important, or I would say all the other system is important. But looking at the heart, if there is a cardiac arrest, no circulation of blood, no nutrients going to circulate to our cells, that means there is no removal of waste and also supply of oxygen and nutrients. Our cell is going to be starved of the needed oxygen. So it basically going to die off. Yeah, that actually is the most dangerous to us. So people generally, when they die, is basically the heart stop beating. Compared to the skeletal muscle, if some of the patient actually lost control of the skeletal muscle due to stroke, okay, due to the problem in the motor system or the nerve, he can still survive. Yes, It's only with difficulties, yeah, that he probably have limitation in the movement. He probably couldn't speak properly. Yeah. Sorry, Hannah. Yeah. Okay, so this we can then look at activity 11.5. You have any question you want to ask about 11.5? No. So we are done with 11.5. Now we look at basic characteristic of the muscles. Yeah. So we look at the role of muscles in homeostasis. Can you give me one example of homeostasis you want to look at? You want to discuss? What internal environment is regulated in us?
Blood pressure.
Blood pressure. Okay, the regulation of blood pressure. We basically look at the acute regulation. Is that all right? Okay, regulation of blood pressure. So we want to regulate the blood pressure. So how is the blood pressure regulated acutely? So we look at the receptors. You know that they are the baroreceptors. So what chemical or what gated channels is present on the baroreceptor cell? What does the baroreceptor detect?
They detect the pressure of the blood, right, doctor?
Yeah, the blood pressure. Basically detect the blood pressure, and you know that they are basically located at the important site, right? So does it has what type of gated channels on the cell membrane of this baroreceptor cell? Anyone? And blood pressure are basically physical, right? Factors.
Oh, is it mechanical?
Yes, because they are physical. So usually mechanical is related to physical factors. So mechanical gate is the one that regulate the physical factors of the internal environment, which is the blood pressure. Okay, if they are chemicals, okay, like what example of chemicals like osmolarity, they are chemicals, then they must have chemical gated or ion channels. Yeah. So this is how you relate to each of them. So now we look at blood pressure. So the receptors is basically the baroreceptors, which have a mechanical gate. So what happens when there is a change in blood pressure? Let's say it starts to decrease. It's a bit difficult. Okay, let's say it start to increase. Yes, Nani. The blood pressure start to.
Cardio acceleratory center is stimulated, right?
Okay, so because the baroreceptor the baroreceptors, they have a certain tone. Okay, basically sending action potential to the heart because we have a certain blood pressure that's around 120 over 60. So so I'm going to on the camera. Okay. Yeah. So you have a baroreceptor here. Okay. So it generally sent to the cardio control center. Okay, and then it's sent to the heart. Okay, you have the heart. There's the effector. Yeah? So the effector, and you have the receptor and also the control center. Okay, so if our blood pressure is 120 millimercury, okay, so basically it has a basic tone in the sense that the action potential that is sent has a certain frequency. Okay, assuming that it meets 120 bit, let's say it send 120 speed of action potential to the control center, telling that okay, that is integrated as 120 millimercury. This is just an assumption, yeah. So as long as the blood pressure is maintained, at least nothing happens. Basically, the channel will be open and it's going to, basically there will be always open of the gated channel that allows sodium to come in. Okay, that generate action potential, and this action potential will be maintained at 120 beats per minute. That's it. Yeah. Because of that, basically it has that frequencies. Okay. Now, if you increase that to, let's say, 125, now this increase in pressure will be detected with more action potential that is being going to the control center. Now it increased to 125. Okay, action potential per minute. So this will then control, this control center will then infer and say, okay, now the blood pressure compared to the error signals, okay, the set point, there's 120. Now there is an increase of 5 millimercury of pressure. Yeah, so it will send then information to the control center to reduce the heart rate, okay, by going through the parasympathetic pathway, parasympathetic system, yeah, to basically reduce the heart rate from, let's say, if it's normally contracting at 70, now probably it reduced to contract at about 68, let's say, beats per minute. Okay, then this is able to then bring back the blood pressure to 120 again. Okay, is that all right? Any question on this? You're clear, right? Opening of the voltage, the mechanical gated channel. Okay, now gated channel for sodium that allows sodium to move in, and also they have a sodium potassium pump that basically moves the thing out. So this is going to be the receptor potential, greater potential, threshold potential, then action potential. And this thing have been maintaining our blood pressure at about 120. Yeah. So if there is any change, either increase or decrease. So if let's say it decreased to 115, okay, now the action potential due to the opening of the gate will basically reduce. So now the action potential that is being sent to here, let's say it's 115 action potential as low. Basically now activate the sympathetic nervous system. Not really, actually it's actually inhibiting, but don't make the thing complicated. Just say that it activates the sympathetic nervous system, then the heart rate increase to, let's say, 72 beats per minute. And that basically then help to bring back the heart rate about 120. Is that okay? So our body blood pressure is basically very closely regulated or acutely regulated. Yeah, which I did not explain much during yesterday classes or the past few day classes, but it acutely regulated so that our blood pressure can be regulated within a very narrow range. Yeah. Okay. Now we can explain the acute regulation. Now, what happens people with hypertension? Okay, if people with hypertension, we know now the blood pressure actually increased to 140 millimercury. Let's say normally it's 120. Now, what happens to this? Why is the heart rate still beat about 70 beats per minute? Can you explain why? Okay, that is the thing's difference now. The receptors, not to confuse you too much, the receptor now actually adapt to the new pressure. So the receptor for people who are hypertension, most of the baroreceptors now actually adapt to the new pressure. And it says now it sends 120 as a new blood pressure that is then interpreted by the control center. Now the control center say, oh, this is the new sector. So basically it sets at 140 ATP. Yeah. So anything above 140 will then open out the voltage, the mechanical gated sodium channel. If not, it will be basically maintained at that. So that's why the blood pressure has been at 120, 140, with the contractions of the heart probably about 70 beats per minute. Is that very confusing? Our body system. Any questions?
No questions, doctor.
Yeah, clear. Yeah. So normally we do not want to get that too complicated, so we just leave that out. So we say that if that is the case, basically now you have 120, so you respond to 120, Okay, so let's say this is 120 ATPs, basically representing. So what does the control center actually read? How fast the frequency that is coming from the receptors, and how slow the frequency coming from receptors will then determine the response from the control center. Yeah. So the control center basically read the electrical signal based on the frequency or number of frequencies or number of action potential that actually arriving to this to be interpreted. Yeah. So this is basically just a reflex that the body normally help to maintain the homeostasis. Yeah. Is that okay? Okay, so we have discussed one role of the muscle, basically the cardiac muscle in homeostasis. Then we already talked about the signal that reach the effector. Yes.
How does the doctor
Yes, please.
to ask how does the control center uses the frequency to identify whether it's
Yeah, because they basically that is the only that it can respond. If you go into more detail about the cardiovascular center, then it has actually many subsections that it actually branch into three. Okay, you have cardiac excitatory center, cardiac inhibitory center, then it has a vasomotor center. I do not want to go into that detail because we are going to do this when we do cardiovascular system later on in your second semester. Okay, then the information that coming will either activates the excitatory or it activates the inhibitory, basically inhibits. Okay, then or it activates the motor center, which basically works on the blood vessels. Okay.
okay, doctor.
Don't worry
Yeah,
at
it's
this
fine
stage.
now.
Yeah. Just basically you understand that basically if there is anything that change in the blood pressure, the mechanical gate is either going to open more or less. That is going to have more or less frequency that is going to your cardiac control center, and then it either basically reduce the heart rate by increasing the activity of the parasympathetic nerve. Yeah. At this stage, just get to know that this is how homeostatic regulating of our blood pressure is regulated. Yeah. The only thing I add a bit more confused to you is that we say what happens during hypertension. People who have a constant high blood pressure of 140, then it's more due to the receptors, the baroreceptors adapt to the change in pressure. Yeah. So it now has a new frequency that is sent to the control center, and that is becoming the norm or the normal pressure for the person who are actually hypertensive. Still okay?
Doctor, does that mean
Yes.
during hypertension,
Yes.
there's a new set point?
Is adapting, is basically the receptors that is adapting to the new pressure. So it also basically reduce. So now in actual fact, the frequency that sent is not basically like 140. Okay, it still send 120 action potential. Why? Because now is the receptor that is adapting, either more gates is open or less gate is open. So this gate, this mechanical gate, adapt to the new pressure. Okay, just like we talked yesterday that about the pain receptors. Okay, after some time, your friends putting hand on your shoulder. After some time, you probably do not feel that your friend's hand is there because the touch receptor basically adapt to the stimulus. Still okay?
All right. Thank you.
Yeah. Okay. Yeah. It's not really a new set point. It's just that the adapting of these receptors, now it still send 120 action potential frequency to your control center. So your control center still thinks that is 120, that is the blood pressure, but in actual fact, the blood pressure has actually gone up to 140. It's a bit confused here, I know. You're okay? If okay, I just move on. If you need further explanation, no.
Okay, doctor.
Okay. So we have done that, and we basically look at how receptor basically know when to contract and when to relax. Okay. So the receptor will know when to contract and when to relax. That is basically influenced by the control center. Yeah. Looking at the variables, it regulates. So it look at the error signals basically the control center is the one that decides looking at the error signals because it's comparing to the set point. So the set point is basically at the control center, and the error signals is actually interpretation of signals, which is also in the control center. Yeah, the error signal is not the one that coming from the receptors. Is that okay? Yeah. Then what conclusion can you make out of this main function? So we thought only on cardiac. So we did not look into how about skeletal muscle. So look at skeletal muscle. How does skeletal muscle contribute in homeostasis? I think you have done some of the exam question. We asked you about if it happens to be in somewhere that is very cold, let's say the temperature you normally have here is around 30, and you get to a place where the temperature went down to, let's say, 0. So what happens? Your body able to maintain the body temperature. Okay, this is where you start to shiver. So shivering is the one that basically your skeletal muscle is contracting and generate heat. And we know that when skeletal muscle contracts, only 40% is basically converted to work. The rest of the 60% is converted into heat. Okay. So question is, if let's say basically if we just sit down quietly in a chair, or if you are lying down on the bed, is there no contraction of our skeletal muscle? Is there any skeletal muscle is contracting? Let's say you are sitting down straight. Is our skeletal muscle contracts at rest?
No.
Okay, so we say they are not contracting. In actual fact, it do contract and maintain a certain posture. So the posture muscle that is contracting, so there must be one that is inhibiting. That means there's one agonistic and one antagonistic muscle contracting to basically maintain your position. Yeah. So if both of them decided not to contract, basically you are going to either fall down one or the other way. So it's important that the skeletal muscle is actually contracting even though we are at rest. And that contraction is the one that basically generates heat. Okay, and that is basically part of our basal metabolic rate that is going on in other cells also basically contribute to the heat. So one of them is basically the skeletal muscle that actually generate our body heat, and that is being maintained at 37 degrees Celsius. Yeah, the normal temperature. Okay, so we talk about skeletal muscle, and we have actually done about the smooth muscle. I just move on. So I believe you have already understood or able to find the answers for 11.6. Okay, so now we move to 11.7, page 20. Looking at the different types of movement that occur in our body. Okay, we have actually done most of it, so I think we can actually move on. Okay, looking at movement, contraction of the different types, and we can see how contraction of the skeletal muscle is initiated. Okay, so I'm going to just write off this and look at how we can initiate a skeletal muscle contraction. So we know very well that the most important thing, let's say this is the skeletal muscle, right? And usually you must have two, okay? And this is the point of insertion in the bone, okay? So it usually have a, let's say the long bone that is coming down and the joint here, okay? Then you have the bones there basically. So there must be a point of insertion on the bones, yeah? And there is basically also another muscles that is, okay? So you have, let's say the tricep, no, not a very good one. Let's say we draw just the long bone in the joint. Okay, so you have the muscles here. Okay, and you have the other muscles that's contracting here. Okay, I hope it look like the bone and basically the hand and the rest of the muscles. Yeah. So this will be the agonistic muscle, the bicep basically, and this is the antagonistic muscle, the tricep. Antago. So you always have both. Yeah. So there must be basically the nerve that is coming down and innervate here, and there is also another nerve that's going to be innervating on the other muscles. Okay, so just put different color pens. Okay, so this is basically the inhibitory nerve. So it's going to basically be known as inhibitory nerve. Okay, but it can be an excitatory. So we just put that as inhibitory. That makes things easier. And then you have the excitatory nerve that is coming down. Okay, at this point. Yeah. So we know at this point there will be a neuromuscular junction that is occurring. So if we activate here, so it must be coming from different neuron, right? So this will be the neuron that is inhibitory, and they are actually coming from one nerve. They're activating both. Yeah. And each neuron excitatory basically releasing acetylcholine here at the neuromuscular junction, and here it must be releasing an inhibitory or GABA, sometimes they inhibit pricing. Okay. So we focus on just the excitatory muscles. So what happens when the nerve coming down? Okay. Now these are the neuromuscular junction. Okay. So what happens here when action potential coming down? Until the nerve endings, the distal part of the nerve endings, what happens here? So there will be basically, is that right? Looking at muscle contractions. So the nerve that basically bring the action potential down is going to open up. We know always that is the calcium channels that is being opened. So you allow calcium to basically move into the cell, right? Then there will be an exocytosis of neurotransmitter. Okay, try to use a different color pen. Yeah. So we have the neurotransmitter, and we know that this neurotransmitter is an acetylcholine. So what will be the receptors here? So what will be the receptors on this surface of the postsynaptic membrane?
Nicotinic cholinergic.
It must be a, yes, it's a nicotinic cholinergic receptors, which is basically an acetylcholine receptors, and this is actually a ligand gated or chemical gated. Okay, so it is known as a chemical gated sodium channels. So it's an ion channels that basically allow. Okay, and we know here is basically the skeletal muscle. So acetylcholine allows sodium to come in. So what changes happens here? In the postsynaptic membrane of a skeletal muscle? What changes happen here when sodium get into the muscle cell?
Depolarization.
Depolarization going to be a greater potential. Okay, so this is sometimes we know as postsynaptic potential. Okay, so it's known as postsynaptic. If they are excitatory, so they are excitatory postsynaptic potential. Right. So it's going to go into threshold potential. So if more sodium coming in now, more sodium actually coming in. So more sodium coming in. So now the membrane potential is going to increase. Okay, from greater potential going to increase to threshold potential. Okay. Now, when you reach the threshold potential, basically there will be an action potential. Okay. Where will be the voltage gated ion channels present on the membranes of a muscle, or sometimes they call sarcolemma? Where will it be present? Most likely. Is here or here? So it's generally will be present about after the synaptic cleft here. Okay, so the greater potential change until about here, so more coming in. So then only the sodium gate is going to open, and this is known as voltage gated sodium channels. Then you have also the potassium gate channels that is going to be here. They allow potassium to move out, and you have the sodium that is coming in. Okay, this is just initiation of action potential. So these two things must occur. So basically we say that now the action potential that happens now, it basically go and spread throughout the whole of the cell membrane or sarcolemma of the muscle cell. Okay, so if you're able to basically spread all over the muscle cell, so if you look at, let's see if I can draw. Yeah, I hope you can see here. Let's say I just draw it microscopic size, the muscle that is coming, the nerve that is innervating. So the action potential is going to spread across the surface of all the cell. Yeah. Okay, so it's going to spread all over the surface of the cell. It's only for this muscle fiber only. It doesn't go into another muscle fiber. So it's only for one, which means that if there are many muscle cells, okay, if let's say you have other muscle cells here, other muscle cells, so each one must be innervated by the nerve. Yeah, so it must be innervated by the nerve. Then you can only allow this to spread across, just only within the cell. Okay, so as this action potential that happens, now we know it has T tubule. This is known as T tubule. T tubule. Okay, the allow action to be potential is basically a continuation of just assuming that this is the muscle, so it's basically continue and pass across, which means the action potential basically just travel down and that's it. It's going out or it's going this way. Yeah, the same thing over here is going this way and it's just going down, which means that it doesn't actually get into the cytosol inside the muscle cell. It's just passing through, but as it passes through, it get to the sarcoplasmic reticulum, which is very closely lined against the T tubule, which is basically a sarcolemma. Yeah, so it's basically very closely lined together. So they are actually very closely in contact of each other. Okay. Then there is also another one that is basically about it. Okay, this is just the initiation of muscle contraction. You haven't started contracting yet. Okay, so this is what happens in our muscles. Yeah, so I just move on to try to answer that. I have not answered how it's going to be basically affect. So the passing over of this action potential will then basically cause the opening of the gate, which is L-type gate, calcium channels that allow calcium to actually come into the cell. Yeah, same thing here. Basically, calcium is released into the cell, and most of this calcium is coming from the store, which is the sarcoplasmic reticulum. Okay, it's actually the same as endoplasmic reticulum, but now sarcoplasmic reticulum becomes the storage space for calcium. So this is where calcium is released. Yeah. Okay, so we have done basically but partially explaining the muscle contraction, and we actually also talk about the muscle looking at activity 11.8. You have any question on activity 11.7? Okay, no. So activity 11.8, we have actually done about heat generation by the skeletal muscle. Okay, and the important thing is because we say that we have about 50% of our body weight is basically skeletal muscle. Yeah, so that is one. If the contraction of the muscle is going to basically generate heat, because contractions, the use out of ATP, basically 40% only contribute to the work, the other 60% basically to generate heat. Yeah. So we have done activity 11.8. Do we have any questions? No question, put out your hand. Okay, so basically no questions, so I'll move on. So we have started looking at how the muscle initiation of the muscle contraction started. So I didn't go into a more detailed explanation. Now we look at the structure of the skeletal muscle. Okay, so if you look through the modules, basically we provide you a lot of pictures looking at how the muscles from the bundles, okay, finally going to become a fibers. Yeah. So from there, you look at the microscopic structures, yeah, the gross structure, and we know the point of insertion and all these things. So these are very anatomical. I leave that to you to explore that. And then we turn to page 24, looking at the microscopic structures. We say that the muscle has cell membrane, and this cell membrane is basically known as sarcolemma. So this is the sarcolemma, the cell membrane, and we know that this is the T tubule. There's invagination of, basically the T tubule does not enter into the cell of the muscle. It passing through the muscles. If there's another muscle, basically it actually go through. Yeah, so it actually passing through. Then they have what we call the sarcoplasmic reticulum, which is basically same as the endoplasmic reticulum. So you can explain the detail on that. Okay, then looking at the myofibril. I think before we go into the myofibril, yeah. So these are important aspects. And if you talk about sarcolemma, so all the voltage gated channel will be outside, away from the synaptic crest or neuromuscular junction. So they are away from that. Yeah, so they are basically present all over the surface of the sarcolemma, of the cell body, of the cell membrane of the muscle. So all the voltage gate will be present here. These are voltage gated ion channels. They are all present. The same thing, they are present here. Voltage gated ion channels along the T tubule and all that. That's why the action potential that is generated by the nerve stimulation basically is going to spread throughout. So if there's T tubule, basically it's going to go down across the cell. Okay. So you will if you see it from the top, basically you see holes on the muscles. Microscopically, you can see holes. So these holes are basically referring to the invagination of the T tubule. Okay, it doesn't break actually, so it becomes like a hole. Okay, so it basically pass through. So you see a lot of holes basically all over the sarcolemma. And then inside the cell, then you will find sarcoplasmic reticulum. Yeah, and this is a site for storage of calcium. Okay. Any questions so far? So if you have none, then we look at activity even more microscopically, looking at the structure of the muscle fiber. So the first one at 11.9, we look at the gross structure. Okay. Then we move on to looking at the microscopic structure. So looking at the sarcolemma, myofibril, all these things. So what is a myofibril and what is it made up of? Okay. The answers provided, you can turn to page 27. Myofibril basically consists of two types of filament. There's the thick and the thin filament, so you can read on that. So I leave that for you to read. And compare and contrast between myofibril and biofilament. Yeah, so you can compare them, and then how they are arranged in the muscle, and also how does that associated to the T tubule. So you can actually look at the figure 11.6, the microstructure of the skeletal muscle. Is that okay? Any question on this activity 11.10? Put out your hand. If no, then I just move on. Okay, no. Anyone response? Yes, so we are moving on, yeah, human. So you move on to the next one, looking at the T tubule. Yeah, the T tubule, you will see that basically it's a bit complicated. It seems like there is a connection So if you look at the structure on page 11, page 26, 11.7, okay, the T tubule, they basically is connected to the sarcolemma. Yeah, so they are basically a triad. So you have the T tubule that is coming down and they are connected to the triad. Yeah, so basically this is basically the sarcolemma. And all throughout the length here, basically there's a lot of sarcoplasmic reticulum. Yeah, so I just leave that not to complicate the whole pictures. So you can see how they are basically aligned to each other. Yeah. So what is the significance of that? And you are asked to draw a voltage-gated calcium channels on the sarcoplasmic reticulum. So if we're going to expand this, so I just make it bigger so that I can draw more things here. So I can just rub this away. Okay. Now you have the T tubule that is coming down here. Okay. So if you draw a huge sarcoplasmic reticulum, let's say this is a huge one, right? And this is the T tubule. If you basically expand that, you can actually see they have ion channels. These are voltage-gated ion channels. Yeah, that's present all throughout, and they are all in arrangement of 4 molecules or proteins. So if you just take one of that, you can see that they are basically arranged in the tetramer. Okay, and same thing over this side. They are also having 4. Okay, so these are basically in combination and they are closely packed together. So if I just rub this off and very close to each other. So I will use a different color pen. Okay, so each one basically they complement to each other. They are close. Yeah. So if I may have to draw this, if you really look at the arrangement, I need to rub this off. Okay, this is to enlarge that. So if you assuming that the T tubule is this way coming down, yeah, I think it's quite difficult for me to draw. They are basically arranged in 4 molecules that are closely packed together. Okay, and they have tubes basically going down. Yeah, and this is basically aligned to those over here on the sarcoplasmic reticulum. They basically align to each other. Okay. Okay. In the way that if there is no action potential coming down, there is no action potential coming down. This is basically the T tubule that is going. Sorry, this is not the right one. Forget about this. Just assuming that I'm enlarging this. Yeah. Okay. So basically the T tubule that is coming down here. So there'll be a lot of these L-type calcium channels. So this is the L-type calcium channels, long-acting calcium channels. Okay, and they are the voltage-gated channels. So when they are activated by action potential depolarization, basically this gate is going to open. There's a conformation change. Is that okay? Yeah. So if there is action potential coming down, there will be a conformation change that allow basically now calcium to be released. Okay, there is also calcium basically coming in, but the one from the outside is not particularly important. But most important, those are coming out from the sarcoplasmic reticulum that is being released into the cytoplasm of the muscles. So the increase in calcium is the most important one that is released from the sarcoplasmic reticulum. So we can draw that, and then if you refer to boron and Barrow, so you can basically look at how this is being transmitted and how it to increase the calcium level in the sarcoplasm, of the skeletal muscle. Did I confuse you? Or you are okay?
Doctor, can you repeat the drawings? This drawing.
Okay, the joints is basically assuming that they are. I just managed to use one to show you. So they are basically closely packed together. So if I can draw it this way, this is a T tubule that is coming down. It has the calcium channels. This is L-type calcium channels. And then they are basically very closely linked to the, can you see that? They are basically very closely linked to the calcium releasing dependent rhododin calcium channels. So they are basically close when there is no action potential, because they are very close together. So basically something like this. Yeah. But when action potential come out, basically there's a conformation change. It moves slightly, basically to open out the voltage L-type calcium channels open. So outside calcium basically also coming. But the most important thing, that opening allow the calcium to be released from the sarcoplasmic reticulum. Can you imagine this way?
Yes, doctor.
Yeah. So this is how it basically, because of action potential, it just moves slightly. So it basically open out. And the calcium release is mainly from the sarcoplasmic reticulum. Even though this is known as voltage-gated L-type calcium channels, those that coming from the external environment or the extracellular fluid is not particularly important. The amount of calcium release is too little to basically cause muscle contraction. So most of this release is actually coming from the sarcoplasmic reticulum. Still okay, Anik? I hope this pen can show how you can visualize that. Yeah, can visualize that. Yeah, and that voltage gate that is present on the sarcoplasmic reticulum is known as the calcium. I just check the names again just to make sure that it's correct. Yeah, it's known as the calcium release channel, and they are also known as rhododin receptors. Okay, so this is actually calcium release channel. It's known as calcium releasing channel. Right. Compared to here, this is L-type voltage gated channel for calcium. Yeah. Okay, this is basically the releasing channel. So the most important thing is because without action potential, they are very closely lined together. But when there's a conformation change in the L-type, it sort of moves slightly, so allow the calcium basically release from the sarcoplasmic reticulum. Still okay? You will take some time to imagine how this thing actually works in the triad. Okay, so we are talking about just one side. This is just one side of the end. The same thing happens over the other side. So this is the T tubule that is coming down. So there will be another triad that is also here. So you will also have this basically present. Okay, the triad. Yeah. That is basically able to release the same thing, the calcium. Okay, over here. Yeah, but because they are very closely together, so it seems to be blocking the releasing calcium channels from releasing the calcium. Yeah. Okay, that's all about activity 11.11. You have any questions? If not, you are confused. You want me to explain again? No. You put out your hand, we can move on. Yes, so we can just move on. Okay, so we move on to activity 11.12. Yeah. Looking at the even more microscopic structures. Okay, looking down to basically looking at figure 11.8a, and then you look at 11.8b, and then you look at, yes, Waka.
Sorry, I have a question.
Sure.
Just to confirm, the calcium releasing channels are the same as the voltage gated calcium channels?
No, they are not. They are different. Okay. The calcium channel is present on the membrane of the T tubule. Is that okay? Which means that it's only present on the membrane of the T tubule. I should draw them nicely again. The whole picture is a bit messed up. Okay, so we have the T tubule that is coming down. I just draw the T tubule that is coming down, right? And you have a big T tubule here. Okay, which means that there's a membrane, yeah, and we make it bigger, okay. So the T tubule. So on the T tubule, basically there will be a lot of the L-type calcium channels. Yeah, I just draw one dot to represent, so there will be a lot of L-type calcium channels. Okay, and they are basically quite closely aligned to the. Okay, this is the sarcoplasmic reticulum. Okay, just to make it, there are two layers. So basically, it's lipid bilayer. Then you will have the channels that will be close together. Okay. I think this is better, right? Okay, so since they are closely aligned to each other, so they basically form a channels. So you have these two together and they basically close. Is that okay, Waka?
Yeah.
Yeah, so they are close. So this is the L-type calcium channels and they are voltage gated calcium channels. Okay, and this is the calcium releasing channels. Calcium releasing channels. Okay, this is L-type calcium channels. Okay, what happens when action potential coming down? So you have this action potential that is coming down. So your action potential that is coming down on the T tubule, basically it spread across. So action potential that is coming down. Okay, so as it comes here, it basically going to activate and open. Basically, it actually move. I just visualize, assuming that it moves slightly internally, slightly. Basically, it opens up. It opens up. That also allow the calcium from the source here to actually move in. So you have an external source of calcium. But I say here, they are not particularly important because they are too little when they get into the cytoplasm of the muscle cell. Okay, so they are not particularly important, so we don't worry about that. But there are other books that explains this. It might probably get you too confused. So we just ignore that. So we don't want to know about this. So the opening of the voltage gated L-type calcium channels do move some of the calcium in, but the amount is too little, so we don't worry about that. The most of the calcium is actually released from the sarcoplasmic reticulum because there's a lot of that is stored in the sarcoplasmic reticulum. Yeah, so the calcium will move out when this one open. So the calcium release increases in the cytoplasm. Is that okay, Waka?
Okay, thank you, doctor. Sorry, I got confused, but I understand.
No. Yeah. So the opening of this is just slightly. Yeah. So allow the calcium to release. If there's no action potential, basically they are aligned to each other. They are close. So it basically the L-type calcium channels is closing the calcium releasing channels. Is that okay?
Yes, doctor. Thank you.
Yeah. Thank you. But some books explain says that the increase in these calcium channels actually open out the the calcium releasing channel, which I disagree because I cannot visualize how it actually opens that out. And we know that basically if you have calcium free, there's no calcium in the ECF. Okay, yet the muscle can still contract, the skeletal muscle can contract. Yeah, because it's innervated by nerve, so it will contract because it does not depend on the external source of calcium. So that answers my question that I asked you just now. Right. I asked you, can the skeletal muscle contract in a solution free of calcium? The answer is yes. Why? Because we do not need calcium source from the solution that's outside, that's the extracellular. Yeah. Is that okay? But we need a lot. That is very important. So the calcium that is released when this move away to open out, so allow calcium to actually come out from the sarcoplasmic reticulum. And that is the most important amount that is released into the cytoplasm that allow muscle contraction to occur.
Doctor?
Yes.
Your question earlier about we put muscle inside a solution free of calcium.
Yes.
Doesn't the calcium releasing channel needs some calcium from the external environment to open at first?
No. Okay, the opening of this voltage gated L-type calcium channels, okay, it also has some calcium that actually comes in. So I said there are some that actually come into here. Okay, that's why I make it small, but the amount is too little to cause a muscle contraction. So if you have a solution free of calcium, it's okay as long as this one open. It's actually releasing the inhibition, or it blocks the opening of the calcium releasing channel. So when this one move away, this calcium releasing channel is open, so more calcium is actually coming out from the sarcoplasmic reticulum. Is that okay, Anik?
Yes, doctor. Understood.
Yeah.
Thank you very much.
Yeah, because some books, as I say, it says that this calcium help to open out these calcium channels, which I don't think I agree. So I disagree with that. So I say they are not particularly important. So I rub them off. Okay, so we don't worry about that. So as long as the skeletal muscle is innervated by nerve, yeah, so it's able to contract in a solution free of calcium. Okay, so I will ask you the next question. That is, can the smooth muscle contract in a solution free of calcium? We are going to get the answer when we do smooth muscle on Monday. Is that okay?
Yes, doctor.
Yeah, that is for discovery, for you to discover. Yeah. So we look at the. Is there any question on this channel, especially on the triad, the transverse tubule and also the sarcoplasmic reticulum system?
Doctor.
Yes.
So basically, for the T tubule, right, the depolarization is spread down the T tubule, right?
Yes.
Okay, so could that mean that the depolarization is just causing the release of calcium ions from the calcium ion releasing channel?
Yes, it's the releasing from the calcium ion releasing channel, which is present on the sarcoplasmic reticulum.
Yeah. Okay. All right.
And
Thank
yeah.
you.
Okay. Thank you. So you can look at figure 11.7a. Okay, it shows how the T tubule basically invaginate or penetrate through the muscle filaments, basically going across it. And also there are orange color, or you say yellow or orange color. Basically, these are the sarcoplasmic reticulum. Yeah. So the TBV is the white color, and the sarcoplasmic reticulum is the orange color. Yeah. The rest of them are the myofibril. Yeah. So this is one strand of myofiber filament. Yeah. Microfilament. You see that in your figure 11.7a? Yes. No. Can someone respond? Do you look at the pictures on 11.7a?
Yes, doctor.
Yes. Okay, good. So when you look at 11.7b, basically this is expansion of that to try to give you the ideas. So now I put it on the drawing on the board. So this is basically how the TT view, this is the red color, and this is assuming that the green color is basically referring to the orange color of the, I don't have the orange color, yeah, the sarcoplasmic reticulum, yeah. So if you are clear on that, so basically that remove a lot of confusion, yeah. So the opening of the L-type calcium channels is also important, but it's important to release the calcium releasing channel. So it's the calcium releasing channel that is the one that actually release the calcium from the sarcoplasmic reticulum. Yeah. So the L-type calcium channels is important to release the calcium releasing channel from inhibition or from blockage. Is that okay? So we are done with that. I think you can work on activity 11.12, basically looking at naming them. And also you can actually refer to a lot of pictures there, looking at the sarcomere. So what is a sarcomere? Okay. If you know the Z line, so it's basically the sarcomere is between two Z line, the middle of two Z line, that is the sarcomere. Yeah. So I remove this. Okay, so if we draw, this is the thin filament. Yeah, basically the thin filament. Okay. So you can draw that, and then have another one. Okay. Then you can draw the thick filament. So the thick filament, basically you can draw then thick. Yeah. Okay. It's not basically hanging in the air. It's actually, there's a spring or a connection, but don't worry about the names. So it's just connecting to the Z. So the Z line, this is the Z line between two Z lines, is called a sarcomere. Yeah? The Z line, yeah, between these two Z lines is basically the sarcomere. So sarcomere is basically one of that, and this is the thin filament, and this is the thick filament. So what constitutes the thick filament?
Myosin.
So this is mainly content of myosin. Yeah. So you are expected there are basically a lot of heads that's coming out. Yeah. So it's actually arranged quite in sequence. Okay. So there. Sorry. So this is basically how the heads actually are being arranged. Yeah. So you can actually draw there, and then you can see the difference. Yeah. So you can learn about the different lines, the I band, the A band, the Z, the M line. So I leave that to you to discover. Yeah. So I'm not going to go into that. Then we look at the thick filament. We already know that basically this is the myosin. Yeah. So I'm going to rub somewhere. Yeah. The most important thing, the myosin must be able to attach to the actin. So this is a bit far away. So if I'm going to basically just, if I can draw one strand, I can basically just draw one. So you expected the head will be basically this way coming out. It's trying to attach to the myosin. So you have the actin. So the myosin head had to be basically almost like 45 degrees. That is able to attach to the actin on the thin filament. Yeah. Okay. So if you basically enlarge the head, so we are going to enlarge the head. Just if you make a, if I can just rub this away here. Yeah. So you basically have a very big head. Okay, and there are proteins basically here, and it has basically the alkaline chain. This is the ATP chain. That's it. This is the alkaline chain. And you have the ATP chain. Yeah. Okay. This is where basically it's going through. Yeah. So they are, if you want to draw the other side, so they basically, there will be another, the head here. Yeah. So you will have the alkaline chain and also the ATP chain, which means that it's actually twisting each other. So there will be another one that's coming down from here, which is probably here. Yeah. And that's basically the head. Okay. So you can imagine that the whole length of the thick filament has all this myosin head that is popping up to try to attach to the actin filament. Yeah. So if you're able to attach and then you release, basically it's going to basically pull. So this is what we call the power stroke. So as the head tilt down, it's basically pulling the myosin towards the Z line. Basically, is pulling here, and this is going to be at the center, the M line. Yeah. Is that okay? So this is the ATPs. that basically works on the ATPs, that it can basically hydrolyze the ATP. Yeah. And the alkaline change, we don't worry about that, basically give it stability. So it basically help the to just like giving the posture. Yeah. So we don't worry about that. Only worry of what happens when it start to work. So we look at the activity 11.14. So you can work the detail. Any question on the activity 11.14? Okay, no, so I just move on. Okay, you can read all the details on 11 on page 32 about the head. So you have the alkaline light chain and the regulatory light chains, which are basically regulating the ATPase activity in the myosin. So this is very important in the regulatory light chain. But when you look at the smooth muscle, this is even more important. You'll have to activate the myosin light chain before the ATPase activity can be increased. Yeah. So then you can look at how the thick filament are different from the thin filament, and then you can do a comparison. So now in the thin filament, there are three types of protein. The contractile protein, that is basically the actin, the troponin, and also the tropomyosin. So you can read that on page 33. But the tropomyosin C is the one that basically binds to calcium. And this calcium basically also probably look at related to the cow modeling on the smooth muscle. We are going to explore that later on Monday on smooth muscle. Yeah. So calciums, when it get into the sarcoplasmic basically get into the muscle cell, is going to bind to the thin filament. Okay. On the thin filament now you have three molecules. There's basically the actin, the myosin, the actin, troponin, and tropomyosin. Yeah. So you can refer to the pictures on page 32. There's a green color, okay? So I leave that for you to explore on that. Yeah? Then looking at the activity 11.15, molecular structure of the thin filament. Is that okay? Unless you have questions. If you don't, I just move on. Yeah. And then looking at the relationship between the actin and the myosin molecule. Okay. So at the rest state, the most important thing is the thick filament is blocked from binding to the thin filament. Basically, the myosin is being blocked from binding to the thin filament. The actin side is due to the tropomyosin. Okay, but on the change the tropomyosin, you have a lot of troponin, and there are three troponin molecules. That is the T, okay, which you can read on page 33, the T, troponin T, troponin C, and troponin I. So you know their functions, yeah? But generally, we just group the three of them together as just troponin. So as long as calcium attached to troponin, yeah, as long as calcium attached to troponin, yeah, please sign out your attendance. Thank you, Akash. You can sign it out now. Okay, as long as you are able, as long as the tropomyosin is being blocked, okay, so there will be no connection between the actin. This is basically at rest. Okay, but what happens during contractions? So when there is a presence of calcium, then the whole activity activated. So you can work on activity 11.17, activation of the troponin and tropomyosin by calcium. Yeah. So if there is calcium then present, so you can, there's a lot of these molecules here. So you have calcium that is coming in. Okay, so the calcium molecule is going to bind somewhere here. Okay, by binding, and this is where over here is being blocked by tropomyosin. Yeah, it's being blocked by tropomyosin. Okay, now when calcium binds to troponin, when it binds to troponin, it actually pulls, or in a way, it actually exposes, it pull away the tropomyosin. Okay, when it expose basically pull it away, then now it expose the binding site here. with the binding site on the thin filament. Yeah, have to use different color. Yeah, somewhere here on the other side, so we have calcium there. So activity of the calcium attached to troponin T, troponin C, that allow the tropomyosin to expose the actin binding site. So for myosin head to bind to it. Okay, so you work on activity 11.17. So okay, so we can work on activity 11.17. So by just binding, what happens? The last question: what happens to the increase in cytosolic calcium? So where is the source of cytosolic calcium in skeletal muscle? Can someone respond? Where is the source of calcium? Yes, Ani.
Isn't it in the endoplasmic reticulum?
Yes, it's actually in the sarcoplasmic reticulum,
Yeah.
which is also related to endoplasmic reticulum. Yeah, so it's actually stored in the sarcoplasmic reticulum. I have explained just now about the triad, about the triads and the importance of it. So basically, that helps to clear out the doubt now. So we know that basically all the calciums that is coming in to the cytoplasm actually come from the internal store. The internal store is basically the sarcoplasmic reticulum. Yeah. Okay, so the increase in calcium, now we know that calcium will bind to troponin C. Okay, troponin C then cause the conformation change. We basically pull or basically move away and basically bring along the tropomyosin. And the tropomyosin then basically able to bind to the actin binding site. Okay, and they are able to bind to each other. Yeah, so we work on other activities, and this is what going to happen. And then we are going to work on activity looking at the page 38, gross anatomical on the musculoskeletal. So we are going to finish that. Unless you have question on page 38, if not, we are going to take a break at almost about 10, and we're going to start back at about 10:30 to look at the skeletal muscle contraction, looking at page 39. Is that okay? If you have any questions you want to ask now before we take a break before we move to the next chapter or muscle contraction. No questions? Okay, so we take a break now. Is that okay?
Yes, doctor. Thank you.
Okay, thank you. So we take a break now and we'll come back at 10:30. Yeah? See, someone asked any question. Okay, so everyone agreed, so we take a break now. We will start back at 10:30 looking at mechanism of skeletal muscle contractions. Okay, so I stop now. We can take a break. Go for a drink.